How's everybody doing? Good weekend? Studied a lot of biochemistry? No. So earlier this afternoon, I did post a grade distribution for the first two exams on the schedule page. I'm not going to put it up here, but if you look under the box that has exam 2, you'll see a link to it, and you can look at it. So any questions, let me know. If you weren't here on Friday and didn't get a chance to pick up your exam, follow me back, and I will give it to you after class in my office. So I'll see you there. All right, so we're moving along pretty well, and as you guys are seeing, it's a lot of material in this course, and it takes a while to get through it. So, but you're bearing up very well. I was very pleased with the performance on the last exam, as I said, so that was very good. What I want to do today is talk a little bit about the immune system. I'm not going to go into too much depth about the immune system. There are complete courses that are offered on the immune system, and we really don't have an awful lot of time, unfortunately, to spend on it. The immune system is pretty fascinating in terms of what it does. And I'll just give you a brief number about that. And one of the brief numbers about it is that our immune system, in theory, is capable of making something like 10 to the 12th antibodies. Okay? So that's 1 trillion antibodies, different immune system structures that can recognize things, which I'll talk about in a second. But 10 to the 12th is remarkable when we consider the fact that the antibodies are made from DNA that makes RNA that makes protein. Okay? So the antibodies are coded in our immune system, but we only have 10 to the 9th base pairs of DNA. Okay? So we have available to us about 1,000 antibodies for every single base pair of DNA that's there. That tells us that there's not simple coding that's giving rise to the antibodies. Okay? The antibodies, in fact, the diversity of antibodies that we can make arise as a result of shuffling of coding sequences. And that's how so many is realized. I'm going to just briefly mention that at the end of the lecture today, or end of the discussion about the about the immune system. But suffice it to say, that's a pretty incredible thing that's happening in the production of antibodies. Without those antibodies protecting us, we we would not be here. Okay, so your immune system is protecting you continuously, and you're probably not even aware of it for the most part. Well, the immune system is a subject that has fascinated people for as long as they have realized that there is an immune system. And it's only been in the past 30 years or so that we've come to realize how, in fact, it is that our immune system is so flexible and so able to meet and match the needs of our bodies. Work on the immune system dates back to the scientist named Jenner, Edward Jenner, back in the 1700s, who was, amazingly enough, a pioneer with a smallpox vaccine. Okay? So when we think of vaccines, we think of very modern type things, but Jenner was actually on top of this as far back as the late 1700s. And Jenner worked and actually had the observation that women who milked cows did not develop smallpox to the same extent that people who didn't, and surmised that they were being exposed to a pox virus that infects cows called cowpox, and wondered if that was giving them some sort of protection against smallpox. And so he actually inoculated women or people with this cowpox, and they found that they did in fact have some protection against smallpox. A pretty remarkable thing. All right, so when we think of the immune system, there are really two main components to it, and they're called the innate immune system and the adaptive immune system. So the innate immune system, as its name suggests, is a component of our immune system that is fairly fixed, and it is there to provide a sort of a constant protection against a constant source of invaders. All right. So, for example, we have protection against invaders, that is, invading cells that have specific structures. Our innate immune system recognizes those, takes them out of action pretty quickly, and we're protected. Now, many of the things that infect us or try to infect us, of course, don't have common standard structures. And for that, we have a component of our immune system known as the adaptive immune system. And as its name suggests, the adaptive immune system is able, in many cases, not all cases, but is able in many cases to adapt to the invader and neutralize it. This happens as a result of action of our immune system cells to recognize that something is an invader and then to make antibodies against it. So this is where the term antibodies first comes up, and it's important to understand that. So I'm going to jump around a little bit on here. Antibodies are proteins of the immune system. They're produced by immune system cells, B cells and T cells. Okay. B cells are what we more commonly think of as antibody-producing cells, but T cells also produce antibodies. What are antibodies? Antibodies are proteins that have a structure that looks like this. Schematically, it's drawn as a Y, and that Y has what are called two long chains or two heavy chains and two light chains. So the two light chains are shown there, the two heavy chains are shown here, and as you see, the two heavy chains are identical, the two light chains are identical. But what do antibodies do? Antibodies recognize and bind to specific structures. Antibodies recognize and bind to specific structures. Those specific structures most commonly are proteins, but they don't have to be. Most commonly proteins, but they don't have to be. So, for example, if you get a vaccine against the smallpox virus, which, by the way, none of you have ever had. That is, you've never had a vaccine against the smallpox virus, which is why people worry about it. Okay? If you get a vaccine against the smallpox virus, your body learns that the protein that's on the coat of the smallpox virus is an invader, and it will make a new set of antibodies against it. The antibodies will bind to that invader and neutralize it. That is, it will prevent it from infecting cells. Okay? That vaccine is very effective. When I was a child, they were still giving that vaccine, and I can tell you from experience, it's one of the more painful vaccines you will ever get because it literally makes you sick. It gives you a very mild case. And you feel achy and creaky and grouchy, and especially when you're a little kid, that's a really bad thing. Okay, so your body is making your body is making antibodies against that. The structure of the antibody, as you see here, there are regions that are relatively constant. Okay, that's the C domain, meaning that from one antibody to the next, this region will be constant. It will not really vary from one to another. On the other hand, at the very tips of the Y, this is what we refer to as the variable region, and these regions vary enormously from one antibody to another one. Okay? It's the variable regions that bind to the antigen. An antigen is something that an antibody recognizes and binds to. Now, this works because antigens have specific structures. You've learned in this class already that protein structure is critical for function. And so invading viruses will have proteins of specific structures. And if the immune system can recognize those structures and bind to them, they can help to prevent infection. Okay? So, variability happens out on the tips of the Y out here. Okay? Now, antibodies are made by cells in the immune system. As I said, we can talk about B cells, we can talk about T cells. B cells are the primary antibody-producing antibodies. Okay? And the antibodies neutralize the invaders. Okay? This schematically shows something about the antibodies. There's a stem cell. This might be your bone marrow, for example, that makes blood cells, might differentiate into other types of cells, as you can see here. Bone marrow giving rise to the B cells, thymus cells giving rise to the T cells. The T comes from thymus. That's where the T comes from. And the B cell comes from bone marrow. Okay? Now, let's see, what was I going to say here? The T cells are in two groups, one that's called helper T cells and one that's called killer T cells. And as their name suggests, the killer T cells are involved in killing invading cells. Okay? So if you had a bacterial infection and it was running rampant in your system, your body would be attacking it with killer T cells. The helper T cells, as the name suggests, help the B cells to make antibodies, to recognize and make specific antibodies. Okay. Let's see, what should I say here? Actually, let's not talk about that. Clonal selection. All right. Now, one of the things we hear about with respect to antibodies are what are called monoclonal antibodies. So it's a term that I want you to be familiar with. And to tell you what monoclonal antibodies are, you need to understand something about how antibodies work and what antibodies are doing. Let's imagine that I am an invading protein in your bloodstream. And I have, obviously, a specific structure. That structure would be globular in nature. You've seen how the structure, for example, of hemoglobin has a specific nature to it. If that hemoglobin were not my own hemoglobin, that hemoglobin would be recognized as an invader because our own system has been tuned not to attack our own cells, our own proteins. And that's a very complicated thing. I won't go into how that happens. But our body is tuned to avoid attacking our own cells. Sometimes that tuning screws up. And if that tuning screws up, then we develop what's known as an autoimmune disease, meaning that we have immune protein or immune cells and antibodies in our body that are attacking some of our own proteins. Autoimmune diseases include things like lupus, very debilitating disease. Rheumatoid arthritis is an autoimmune disease. And psoriasis, skin disorder, is also an autoimmune disorder. Okay? So these happen when your immune cells are attacking things in your body. Now, when the immune cells, when the antibodies and the immune cells are attacking a target, they are attacking only a portion of a target. Only a portion of a target. So if I have a hemoglobin, we could think about that hemoglobin had sort of a donut structure. And we recall that hemoglobin had 4 proteins that were in it, 2 alphas and 2 betas. Okay? We might imagine that an antibody that was recognizing, let's say, a dog hemoglobin that was in my body would recognize maybe a portion of the alpha alpha globin protein in the hemoglobin. So one part of that. The part of a protein or the part of a structure that an antibody recognizes within an antigen, so we're talking about within an antigen, is called an epitope. E-P-I-T-O-P-E. All right? An epitope is, you can think of as a subsection of an antigen. So the antigen is the overall protein that's being recognized. The epitope is a specific region that's being bound by a specific antibody. Well, that tells us that for a given protein, our immune system might make several different kinds of antibodies, each recognizing different portions of that protein. Each recognizing a different portion of that protein. Now, if that's the case, we have what's called a polyclonal antibody. And our immune system makes polyclonal antibodies. That is, it makes many different antibodies, all of which recognize the same antigen, but different parts of it, different structures within that antigen. We got plenty of antibodies because remember we can make 1 trillion of them. So our immune system might throw a couple hundred antibodies against a given antigen. All right? Well, what's a monoclonal? A monoclonal antibody is something that we can make in a laboratory that recognizes and binds to only one specific epitope. It recognizes only one specific thing. Now, monoclonal antibodies turn out to be very, very powerful tools, both for the purpose of research and for the purpose of medicine. Okay? There is a monoclonal antibody that's very commonly used in treating breast cancer, known as Herceptin. You don't need to know the name. All right? And Herceptin is a monoclonal antibody that recognizes a protein that's found too abundantly on breast cancer cells. Okay? It's found too abundantly on breast cancer cells, and this monoclonal antibody attacks it and stops the protein from telling the cell, divide, 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 which is what it's doing when it's present in too great of an abundance. Okay? So monoclonal antibodies can be very, very powerful tools for helping us to treat disease, as well as used for studying in the laboratory. Okay. This shows, again, how there's the antigen is this sort of fluffy cloud-like thing that's there, and it shows the antibodies that are binding to it. We can imagine that, again, this antibody here might be recognizing a specific structure here, the same structure over here, whereas this antibody is recognizing a structure over here versus a structure here. A given antibody is always going to recognize the same structure, but different antibodies will recognize different structures. Okay. Now, the last thing I'll say about antibodies in general is that antibodies come in there are 5 different categories of antibodies. Okay? I'm not going to give you the names of them, but suffice it to say there are 5 different categories of antibodies, and they have different interactions that they do. Some have more than just this Y structure that we have here. The Y structure is the most common structure that we see in antibodies, but there are others that have other structures as well. Now, I said I would tell you a little bit about how antibody diversity arises, and I want to do that, and I don't have any figures for that, unfortunately, so I'm going to do that, and then I'll finish talking about antibodies. So, antibodies are coded for by cells, the immune system cells, the B cells, for example. So we're talking about a B cell has DNA within it that codes for antibodies, and that those B cells' DNA has to be able to code for, theoretically, any given antibody. Well, how does it accomplish what it does? They said there's a certain shuffling of the genetic information that allows this to happen. Okay? When I tell people that every cell in your body has the same DNA, I lie a little bit. Because your immune system is one place where your DNA is actively being rearranged in order to produce additional antibodies. Okay? So there are rearrangements that happen inside of your immune cells that are shuffling coding sequences that make antibodies. Okay? Shuffling coding sequences that make antibodies. We talked about how eukaryotic proteins are in pieces, and those pieces are called exons. And one of the common questions people have is, well, why are eukaryotic proteins in pieces? And the simple answer is that the shuffling of exons over evolutionary time is much easier if they are, in fact, in pieces. That is, the evolution of proteins happens faster if segments of the protein rearrange than if we try to rearrange intact proteins. It doesn't happen so readily if we try to do that. Well, what immune system cells are doing is they're actually evolving antibodies as you're sitting here listening to me. They're evolving antibodies. And they're doing this by two primary means. One is they're rearranging their DNA to position exons around each other in different ways. The other is that they're using the phenomenon of splicing. And splicing, you remember, involves cutting out regions of introns and joining exons together. Depending upon how we splice things together, we can make a lot of diversity depending upon what things end up in that final messenger RNA. Now, the important thing is whatever mechanism a given cell is using, once it has been stimulated to start producing what it currently has, it's then stuck in that mode. It's not going to change after it has differentiated to the point of making a specific antibody. And then it's going to make a lot of those antibodies. And it's going to be stimulated to make those antibodies by helper T cells. The helper T cells will be out there saying, hey, I have found an antigen. I have found an antigen, and I'm going to interact with this B cell, which has also found this antigen. And once the T cell recognizes the B cell has found the antigen, it says, you're the one I want to divide. And so that B cell will be stimulated to divide, and make lots and lots and lots of antibodies. Okay? So that's what's happening during immune stimulation. Well, how do we get immunity then? All right. Well, once we have stimulated a given set of antibodies to be produced, our body has a memory associated with it. Our immune system has a memory associated with that. And that memory is good for about 10 years or so. That's why whenever you get a tetanus shot, if you haven't had a tetanus shot in 10 years, you should get another one because its effects will wear off because the memory of making the antibodies against that tetanus organism, all right, is lost. But if you re-stimulate it, then again, the immune system will say, oh, yes, okay, here I'm going to make antibodies against this. I had smallpox vaccine when I was about 6 or 7 years old. I haven't had one since then because they don't give them since then. I have no more immunity to smallpox than you do because my immune system has long since forgotten the fact of how to make the antibodies against the smallpox virus. Okay? All right. Now, that's kind of a mouthful of things. But suffice it to say that this shuffling of exons via splicing or via recombination allows for an incredible number of different antibodies to be made, just like shuffling a deck of cards gives an amazing number of possible arrangements of cards. Exactly the same phenomenon that's going on. Okay, that's what I want to say about the immune system. Any questions about that? No? Yes? I'm sorry? Yes. So it's shuffling sequences to do that, but once it has differentiated to make a specific antibody, then it's not going to shuffle anymore, and it's not going to splice any differently. It's going to continue with what it's got and make the same antibody over and over and over. Yes? Yes. Okay, so exons and introns. Exons are the coding regions in a eukaryotic messenger RNA that get joined together during the phenomenon of splicing. So it's introns, they're the parts that get removed. Exons are the parts that get joined together. So exons are coding regions in an RNA. I apologize if I wasn't explicit on that earlier. Yes? Women, I think you're talking about the monoclonal antibody I talked about for breast cancer, right? That's Herceptin is what it's called. Herceptin. Is that the question? Or? Oh, Jenner. He noticed that the women who milked cows had a lower susceptibility to getting smallpox, and it wasn't a chemical. He found that they were being exposed to a related virus called cowpox, And cowpox was giving them some immunity against the smallpox. And so it gave him the first clue that there was some sort of protective effect of exposure to a disease. There had also been hints before that people who had survived smallpox were more likely to also be protected against a future infection. So those sort of went hand in hand. Okay, that's enough for the immune system. Let's turn our attention. We're rapidly moving in the direction now of metabolism. And before we talk about metabolism, we're going to talk about two things. One is I'm going to talk about energy. So we're going to have a little bit of calculations on the next exam. Not a lot, but a little bit. And then we'll talk about structure, and then we're going to be headlong into metabolism. All right. Well, I promised when we started the term that I would come back and talk about energy at some point, because energy is absolutely critical for us to understand, because energy determines whether or not chemical reactions occur. Energy ultimately determines that, okay? You know what the Gibbs free energy is. You've heard about the Gibbs free energy, and the Gibbs free energy, I'll remind you, is a measure of energy that ultimately can be used to predict the direction of a chemical reaction. That chemical reaction can be catalyzed by an enzyme or not catalyzed by an enzyme, and the energy is exactly the same. All the enzyme does is it speeds the reaction. Okay? Well, in the first exam, we talked about the Henderson-Hasselbalch equation. And you didn't have to memorize it, but you probably got tired of hearing me say pH equals pKa plus log of salt over acid. Okay? Here's another equation you're not going to have to memorize, but you're probably going to hear a lot of, and that is delta G equals delta G0 prime plus RT times natural log of products over reactants. Okay? These two equations, very, very similar. Okay? And it's for this reason that I think people find that doing energy calculations are pretty simple, because they learned a lot in doing Henderson-Hasselbalch type equations. In Henderson-Hasselbalch type equations, pH was something that varied depending upon proton concentration, right? In energy equations, delta G is something that varies depending upon the concentration of things that we use, very much like the pH was with respect to proton concentration. In the Henderson-Hasselbalch equation, the pKa was a constant for a given system, a given buffer, right? So if I had an acetate buffer, its pKa was 4.76. In the Gibbs free energy equation, delta G0 prime is a constant for a given reaction. It's a constant for a given reaction. For a given reaction, it will always have the same value. R is simply the gas constant R. It's just a number. It's a constant. T is the temperature, and in this class, we won't vary temperature. We'll always use 300 degrees Kelvin. Okay? And now we have concentration of products divided by concentration of reactants, just like we had concentration of salt divided by concentration of the weak acid. This equation is very, very similar. Instead of a log term, which is a log base 10 term, we have a natural log. But the principles are still the same. That is, if this ratio is greater than 1, the log term is positive. If the ratio of products to reactants is less than 1, the log term is negative. Exactly like we saw with the Henderson-Hasselbalch equation. Okay? Now, so these are very important things for us to keep in mind as we're moving forward. Now I'm going to give you some examples, and I'm going to walk you and talk you through a couple of these. But again, remember, I like to think of these in practical terms, not in I'm going to punch this into my calculator terms. I'll wager that none of you used your calculator on the first exam, because that's how I structure my mathematical problems. Okay. The delta G, this term right here, okay, delta G, the top term on the left, is the most important consideration for predicting the direction of a reaction. You probably learned in basic chemistry in your freshman year that if the delta G for a reaction is negative, the reaction goes forward as it is written. If the delta G for a reaction is positive, the reaction goes backwards as it is written. And if the delta G for a reaction is 0, the reaction is at equilibrium. Now, as I mentioned earlier when I talked about this equation, equilibrium does not mean equal amounts of products and reactants. Get that in your head. Okay? Equilibrium simply means that the forward reaction is going as fast as the backwards reaction. There might be 10 times as much product as there is reactant. All right? But if the delta G is 0, it's at equilibrium. It's not changing in concentration of products and reactants over time. Okay? Very important thing to remember. And about a third of you are going to get on the exam and you're going to tell me that delta G being 0 means that products equal reactants. And you're going to be wrong. Okay? So save yourself the embarrassment, save yourself the points, get out of your head the fact that equilibrium means equal concentrations of products and reactants, because it doesn't. It means the system is at equilibrium. That's what delta G0 means. Okay. Well, let's think about this. Let's say that I have a reaction that has a delta G0 prime of plus 4 kilojoules per mole. Okay? At equilibrium, what can you say about the ratio of products to reactants? Are there more products or are there more reactants? Products? Anybody say reactants? Somebody should have said reactants because there's more reactants. Why? How do I know that? Well, at equilibrium, what do I have? Delta G must be equal to 0, right? Delta G is always 0 at equilibrium. 0 equals 4 plus this thing over here. That means that this thing over here must be equal to negative 4. How does this thing over here become negative? If the ratio is less than 1, that means I have to have more reactants than products, right? Right? Okay. Let's say that I have a reaction that has a delta G value of plus 3. And there are equal quantities of products and reactants. What can you say about the delta G0 prime? So delta G is positive 3, and there's equal quantities of products and reactants. What can you say about the delta G0 prime? It's equal to 3, because equal concentrations of products and reactants means that the log term must be 0. There's where equal means 0, not over here where equal means 0. Okay? Equal concentrations of products and reactants means this log term is 0. So if this is 0 over here, then 3 must equal what delta G0 prime is. Okay? Delta G0 prime is called the Gibbs standard free energy, or the standard Gibbs free energy. As I said, it is a constant at a given, for a given reaction. It's a constant. It will never change for a given reaction. Okay? The delta G0 prime is called the delta G0 prime because it is a Gibbs free energy. It's the Gibbs free energy at standard conditions. And standard conditions include 1 molar products and 1 molar reactants. You begin to see the pattern here? These two being equal, this term ends up being 0, and these two guys are equal. So delta G0 prime is the change in Gibbs free energy when we're at standard conditions. Okay. Now, I'm going to recommend that you work through some of the problems that are there that I provided for you. You'll see there's some videos of me working those problems as well. As you'll see, I'm not hung up on what the numbers are, but I am hung up on your understanding what's happening in those reactions and what you can tell me about this without using a calculator. Okay? It's okay to use a calculator to go through those if you wish, but you can tell me most of those unless I'm asking you for a specific number in the problems. You're not going to need a calculator. I'm not going to ask you for a specific number on an exam unless you can do it without a calculator. Okay? Okay. Questions or comments? Monday classes are always the least interactive. All right. Well, why do we worry about energy? Energy is important because energy is necessary for life. Energy is necessary for life. We hear that, of course, entropy is chaos, and everything tends to want to mix up. But I gave you an example on the first day of class about how if we expend energy, we can, in fact, organize things. And that's what our cells are continually doing, is they're organizing things. To organize things requires energy. It's why we have to eat. It's why plants have to absorb sunlight. It's why any living system has to have something it can exploit as an energy source. Okay? Ultimately, all of the energy on the face of the earth comes from the sun, and that feeds plants with photons of light. The photons of light in the plants, as we will see, are used to make glucose and produce oxygen, which is what we breathe in our atmosphere. Okay. Glucose is a storage form of energy that is available to us when we need it, and glucose, as we will see, is wonderful for rapid release of energy. That's the primary function of glucose, rapid release of energy. Your body can dump glucose into the bloodstream in seconds, get it to the muscles in seconds. Your muscles have stores of glucose in the form of glycogen that they can break down in seconds or even less than seconds to have a total burst of energy, very rapid, to help you to run away, escape, do what you need to do. Okay? Fat doesn't work that way. Fat is much more complicated and much more difficult to move in the body. All right? Well, we're making glucose and we're burning glucose to use to make the fuel that does all the stuff for us, and that fuel is ATP. ATP is necessary for moving. ATP is necessary for thinking. ATP is necessary for making things. And as our body is dividing, our cells are dividing in our body, or we're making immune cells, or we're making antibodies, or any of these sorts of things, it takes energy to build them up, just like it takes energy to build a car or to build a house. Okay. Two terms to be very familiar with. One is catabolism. Catabolism. It's up here on the upper left. Catabolism is a process, is a metabolic process that breaks things down. It takes big things, breaks them down into smaller things. Catabolic processes are oxidative. They are oxidative. Things get oxidized as a result of catabolism. When we oxidize glucose, we produce carbon dioxide. That's an oxidation process. Oxidation processes are energy releasing in general. They release energy. Okay? The other term to be familiar with is anabolic or anabolism. Anabolic processes are exactly the opposite. Instead of being oxidative, they are reductive. Instead of breaking things down, they are making things. They take small building blocks and they build them into bigger things. The synthesis of a protein is an anabolic process. And because they are building things, they are energy requiring. Okay? So anabolic processes are the opposite of catabolic processes. Okay. So we take nutrients into our body in the form of fats, polysaccharides, and proteins. They provide us with energy, they provide us with storage materials, and they provide us with building blocks. All of these are necessary for us to be able to take these things and then build them into something that's useful for our cells. So that we get energy from these guys. We also get building blocks because amino acids, you know, we have essential amino acids that we have to have in our diet. They're coming, obviously, from our diet. Okay. I'm talking about oxidation and reduction. It's important to understand what oxidation and reduction means. Oxidation does not mean oxygen. It can, but it doesn't need to. Oxidation means that something has lost electrons. In this scheme you see on the board, or on the screen, ethanol is becoming oxidized as we move from left to right. It's becoming acetaldehyde. In the process, ethanol is losing 2 electrons and 2 protons. If I were to run the reaction backwards to the left and add 2 protons and 2 electrons to acetaldehyde, I would be going through a reduction. From left to right, I'm going in a catabolic direction. From right to left, I'm going in an anabolic direction. Okay? So, if you want a definition for oxidation, oxidation always means loss of electrons. Reduction always means gain of electrons. For every oxidation, there is a corresponding reduction. Something that loses electrons here is giving them up to something else over here. Something else that gains these electrons will become reduced in the process. Okay? Now, we will see this happen over and over in metabolic processes because our cells do not oxidize things and let those electrons go flying away free. Very good reason not to. Those electrons will cause molecules to become, that they attach to become extraordinarily reactive. We don't want that to happen. So we have what are called electron carriers that grab those electrons and keep them from being, or keep them from creating reactive compounds. Okay. One of the carriers is known as NAD. Okay. One of the carriers is known as NAD. No, you're not going to draw the structure of that. But suffice it to say that the electron carrying portion of NAD is up here at the top. Okay? You see here's a nice benzene ring, and you can see over here it's been changed. Okay? This guy has gained a hydride ion, meaning it's gained 2 electrons and 1 proton to make this structure on the right. Okay? On the left, we have something called NAD+. On the right, we have something called NADH. Why doesn't NADH have a plus beside it? Well, over here, this guy had a plus charge. Over here, it gained a proton and 2 electrons. A proton has a charge of +1. 2 electrons have a charge, each have a charge of -1. So we have a +1, +1, -1, -1. NADH has a charge of 0. Okay? There's a related compound called NADP, or NADP+. And by the way, I frequently won't put the plus there. I'll just call it NAD, but it's always implied that NAD, when I say NAD, that is a plus. And the same thing is true of NADP. NADP+, always, the plus is always there if you're talking about NADP. Okay. NADP is a related compound, and it's made by putting a phosphate onto that position right there. These compounds, even though they have similar structures, have fairly different functions inside of cells. NAD is used in catabolic processes, the product of which is NADH. NAD is gaining electrons. It's becoming reduced because something else got oxidized and gave up those electrons. If I had NADP and it gained electrons, it would become NADPH, and it would also have a 0 charge. And NADPH is used primarily in anabolic processes, that is, making something. NADPH is very important for making fatty acids, for example. We'll see some other examples as well. Another electron carrier is known as FAD. FAD looks like you see there. And FAD, instead of accepting 1 proton and 2 electrons, it accepts 2 protons and 2 electrons. It goes from the oxidized form on the left to the reduced form on the right. You'll notice its charge is 0 here, and its charge is 0 over here, but its structure has, in fact, changed. And let's see. And there's FAD. Riboflavin, you see right there. One of the reasons you need to have riboflavin in your diet is so that you can make FAD. Very important. And a good reason for eating chocolate, by the way, is the fact that chocolate is full of flavins. And flavins are good for you. Everybody's happy now, right? Okay. I earlier talked about phosphates and the fact that phosphates are energy storage forms inside of cells. And one of the reasons that they're energy storage forms is because they repel each other. This guy here has got a negative, a negative, and a negative, and they're all very close to each other. And to get those 3 negatives very close to each other, you have to put energy into them. There's energy stored in each one of those bonds in order to make this guy be stable. Okay? That means that if we break those bonds, that energy is released, and that energy can be used for something. That energy is used for everything that we do. Okay. Not much more to say there. Okay. Some of these compounds that are phosphate containing have incredibly high energy. Okay. Here's the energy that is in the form of ATP. Okay. ATP has a delta, that is the breakdown of ATP to ADP, has a delta G0 prime of minus 30.5. I'm coming back to delta G. Okay. If the delta G0 prime is equal to minus 30.5, okay, and I have equal concentrations of products and reactants, which way is the reaction going to go? Forward, meaning that ATP is going to break down into ADP. Minus 30 is a very high value for delta G. That means it goes bang. It really goes forwards. Okay? There are compounds in the cell that have higher potential energy than ATP. Okay? Acetyl phosphate, creatine phosphate. This is in your muscles. Carbamyl phosphate. Phosphoenol pyruvate. We'll see that in glycolysis. It's called PEP, P-E-P. Has about twice the energy of ATP itself. Well, how do we make something that has more energy than ATP does? You may find this hard to believe, but we can actually use ATP to make something that has more energy than ATP does. How can we do that? What's that? Use more than 1 ATP. It's not really an option, unfortunately. Nice guess, though. Let's go back to the equation, and then I'll finish with this. Okay? Here's our equation. What determines if a reaction goes forward or not? Let's say I've got creatine plus ATP goes to creatine phosphate plus ADP. I can only use 1 ATP. How do I make that reaction go forwards, even though the overall delta G0 prime would be positive? I make a lot of reactants. Exactly. Very good. Okay. By putting a lot of reactants in there, I make this term be more negative than this term. I can make it go forwards. So though technically I can't use more than 1 ATP to make the molecule, I can certainly put a lot of reactants in there and make it energetically favorable to make that molecule. Okay, that's a good place to stop. Let's stop for today, and I will see you tomorrow. Oh, by the way, I do have a song if you want to sing a song. I almost forgot. Okay. It's to the tune of Danny Boy again. It goes. O delta G, the change in Gibbs free energy, can tell us if a process will advance. 'Cause if the value's not less than naught, it translates that reverse reactions haven't got a chance. But when the sign is plus, it is the opposite. And then the backwards happens all the time. A factor is the standard Gibbs free energy. So don't forget about the delta G naught prime. Okay.